my name is Madeline, and this year my project is trying to use insulinoma, a benign pancreatic tumor, in order to create a new solution for people with diabetes. This project started when I wanted to research islet cell transplantation, a current way to mitigate diabetes. I wanted to test the potential of insulinoma of replacing isolates in transplantation. I plan to test this 2026 next year. The goal of this project is to find the most viable human-animal model for an insulinoma transplant. I chose to use C57BL6 and 129 laboratory mice. I tested these mice using glucose test, CA199 test, and Ki67 biomarker tests. All testing of mice and equipment were used by my designated scientist in a university lab for safety reasons. Results were compared to compiled data from an external study in order to see what glucose levels trends correlated with successful islet cell transplantation. Now, why did I choose these tests? The glucose test identifies successful islet cell transplantation, the CA199 test identifies cancer risk, and the Ki67 tests are a staining test that identify rapidly dividing cells. For the CA199 and Ki67 tests, supply constraints delayed this part of the testing, and I will be presenting the data at the state science fair. From my glucose test, I was able to conclude that the C57BL6 mice would be the best human-animal model for insulinoma transplantation because they had, on average, 11% lower blood glucose levels. Why are low initial blood glucose levels important? It alleviates the workload on the newly transplanted cells and allows for the healthy endocrine system to compensate for the workload. This conclusion is supported by all my data and statistical analysis. When I first thought of this idea in 8th grade, I never thought I would actually be able to experiment on a potential diabetes cure. Now I believe that this kind of research is reusing discarded tumors and can extend the frontier of modern medicine in diabetes and countless other diseases. Thank you so much for listening. Please see my board for more details.